Assalamualaikum warahmatullahi wabarakatuh. My name is Nakir Fakri Nazri and I will present our introduction. Okay, first of all, microalgae biomass has a faster growth rate, higher biomass production, and higher biomolecule accumulation. So it is making it as an essential feedstock for biofuels and other bioactive substances. In order to increase this biomass productivity, this study sought to maximize the specific growth rate of the freshwater green microalgae, which is Clostridiopsis acicularis, a member of the Chlorellacea family. Conversely, microalgae is an attractive substitute of the traditional materials due to preferentially high productivity, faster growth rates, less land requirements, less harvesting time, and simple nutrients requirement for its growth. Algal cells are mainly rich source of lipids, carbohydrate, proteins, and other value-added products. Several algal species rich in total carbohydrates, such as Chlorella sp with 77.6% and Sandesmus sp with 58.6%, which are reported in previous studies to be suitable for biofuel production. Being rich in cellulosic content, algal biomass offers advantages over other biofuel feedstocks as it biochemically algal cells lack in hemicellulose and lignin, which make it simplifies the process of bioethanol production. Freshwater green microalgae, which is Clostridiopsis acicularis, was isolated and characterized for high specific growth rate and biomass productivity with aim to be used as bioethanol feedstock.
Assalamualaikum, my name is Muhammad Zulukman. I will talk about the production process. Sampling collection. Freshwater microalgae samples were collected from Rawal Lake, Islamabad with the aim of isolation and identification of potential strain people of growth faster and biomass productivity. Growth condition. An antibacterial and antifungal concentration of 0.1 microgram per milliliter each is added to enrichment media prior to sample transfer in order to avoid any fungal or bacterial contamination. Modified cetyl trimethyl ammonium bromide method was used to separate genomic DNA from algal cells. 0.25 gram of harvested algal cells was grounded and dissolved in preheated at 65 degrees Celsius 1.5 ml of cetyl trimethyl ammonium bromide buffer. Central Composite Design (CCD) of experiment was used where pH and phosphate concentration were considered as independent variables in order to optimize growth characteristics. The significance of linear, square, and two-way variable interaction and response of specific growth rate and biomass productivity has been estimated using analysis of variance, which is ANOVA. For growth characteristic, the specific growth rate and biomass productivity of microalgal cells were evaluated by measuring their dry weight on day 3 and 21 respectively. Early and late expansional phase of cultivation. Cellular pellets were oven dried after being washed twice with distilled water and centrifuge at 4000 rpm for 12 minutes. Biomass analysis. Analysis was carried out in FTIR spectrometer fitted out with zinc selenide ATR detector with the resolution of 1 cm and 15 scans in range of 4,000 and 600 cm. The quantity of carbohydrates, lipids and proteins was assessed and compared by determining the absorbance intensity of spectral peaks assigned to each biomolecule. Saccharidification and submerged fermentation. Saccharidification was carried out by acid-catalyzed hydrolysis of biomass. 500 mg of oven-dried and finely ground biomass was subjected to 2% H2SO4 solution and autoclave for 15 minutes. Yeast extract peptone, dextrose medium was used to activate the yeast cells. Lastly, estimation and characterization of bioethanol. Solvent extraction and dichromate oxidation method was utilized to measure concentration of bioethanol. 15 grams of potassium dichromate would dissolve in 100 milliliter of 5-MH2SO4 solution. Tributyl phosphate, which is TBP, was used as bioethanol extractant. Sample were mixed with TBP in ratio of 1:1 in 2 milliliter Eppendorf tubes and vortex vigorously until two phases separated out. Optical densities of solution were measured at 595 nm in UV spectrophotometer.
Traditional bread making technology relies on ingredients such as wet flour, yeast, water and salt to produce bread. This method has been used for thousands of years and has evolved over time to produce different varieties of bread. Yeast that used in this method can produce carbon dioxide. In contrast, the use of microalgae in bread making is relatively new technology that seeks to improve the nutritional profile and sustainability of bread production. Microalgae are a type of single-cell organism that can be grown in a large quantity using sustainable methods such as algae-based system or photobioreactor. They can be processed into a flour-like substance and used as ingredient in bread making. The key difference between the old and microalgae technology is the source of ingredients and the method of production. Traditional bread making technology relies on the traditional wet flour, while microalgae technology utilizes microalgae as a novel source of protein, fiber, vitamin, and mineral. The use of microalgae in bread production also offers the potential for a more sustainable and environmentally friendly bread making process.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Danish bin Abdul Aziz. Next, I'll move on to the profits. Microalgae have a high growth and are able to produce large amounts of carbohydrates, which can be fermented into a bioethanol. Unlike traditional crops used for biofuel production, such as corn and sugarcane, microalgae can be grown in a variety of environments, including saltwater and wastewater, reducing the competition with food crops from land and water resources. The process of producing bioethanol from microalgae can be more efficient and cost-effective compared to traditional methods, as a microalgae can be produce more biomass per unit area and do not require the use of energy-intensive inputs such as fertilizer and pesticides, which the increasing demand for renewable energy resources. Investing in the production of bioethanol from microalgae can be provides a substantial return on investment. That's all from our group. Thank you.